Chapter 16, Disease and Epidemiology. Epidemiology is the study of the distribution and determinants of health-related states or events, and the possible control. It's like the how, when, and where infections occur. Let's start out with some terminology. Pathology is the study of disease. Etiology is the study of the cause of a disease. Is it caused by contaminated food or water? Is it caused by mold growing in the walls of a house? Is it caused by a bacteria or a virus? It looks at the cause of the disease. Pathogenesis is the development of the disease. How does it progress, the development of it? Infection is the colonization of the body by pathogens. Remember, not all microbes are pathogens. Our body can be colonized with normal bacteria that's good for us. But when it causes an infection, it's colonization of the body by pathogens. Disease is an abnormal state in which the body is not functioning normally. There's something that's happening that is abnormal and causing the disease state. To have a very healthy normal microbiota, a very healthy normal microbiome is really important for our health. Our normal microbiota can help to prevent the colonization of pathogens in or on our body. We have a normal microbiome on the surface of our skin. We have a normal microbiome within our body. Transient microbiota may be present for days, weeks, or months. It's only a temporary type of microbiota. It is not permanent, but we do have that normal microbiota that is permanently colonized on our body and in our body. It permanently colonizes the host. Symbiosis, when we're talking about the normal microbiota, is the relationship between normal microbiota and the host. And remember, when we think about these relationships, they can be positive or they can be negative. We need to remember what those terms mean. Commensalism is when one organism is benefited and the other is not affected at all. Mutualism is when both organisms benefit from the relationship. Parasitism is when one organism is benefited at the expense or the harm of the other. Think about that relationship between bacteria and viruses. A virus needs a host. It can get into a bacteria and replicate and make more viruses, but it harms that host cell because it destroys that host cell sometimes when it lyses it in the lytic cycle. So one organism would benefit, the virus, at the expense or harm of the other, the bacteria or host cell. So that's what parasitism is. Now, some of our normal microbiota can become opportunistic pathogens. Sometimes when our body is under stress, when our immune system is weakened, sometimes when there's trauma, our normal microbiota can become opportunistic and cause an infection. And we will talk more about this. I like this image because it shows the different portals of entry where pathogens can gain access to the body. In our eyes, in our nose, through broken skin, through a parental route, through a hypodermic needle, through an insect bite. It can enter our bodies in many different ways. We've talked a little bit about the portals of entry. What is good to note is that wherever these pathogens can try to make entry into the body, we have a normal microbiome in those areas that's like a first line of defense. It's the first encounter for that pathogen. And if our normal microbiota is healthy and strong, sometimes we can keep the pathogens out of those areas by competition, by preventing the ability for that pathogen to find a place to take hold. So having a good normal microbiota is really important for us, and it is throughout our body and in our body. Our normal microbiome can protect us through competition. That competition means that we have a normal microbiome of bacteria. We also have a normal microvirome. We have a virome that can protect us against pathogenic bacteria taking hold within our bodies. It does this through antagonism. Microbial antagonism is competition between our normal microbiome and the pathogens that are trying to infect us. So the normal microbiota protects the host by occupying places where the pathogens want to attach. If the normal microbiome or the virome are present, then those pathogens don't have space to attach within our bodies. They can produce acids. It produces acids that bacteria can't tolerate. Some bacteria do not like an acidic environment. So our normal microbiome can protect us by producing acids. They can also produce antibacterial chemicals. So that also helps to prevent the pathogens from taking hold within our body. Now, probiotics are live microbes applied to or ingested into the body. You might have heard of people who take probiotic tablets to boost their immune system, that boosts their normal microbiome. Or you could eat yogurt that has a good, healthy dose of probiotics. Or fermented foods also have probiotics that can be very beneficial to our normal microbiome. Koch's postulates have been around for a long time, and they're still used today. They're used as a frame of reference, and you can go through the steps in order to trace back an infection to its source. It's used to prove the cause of an infectious disease from a specific microbe. So the first step, the same pathogen must be present in every case of the disease, but it is not found in a healthy organism. So you would look at samples from infected individuals and you would compare them and you would see if they were the same pathogen. Number 2, that pathogen must be isolated from the diseased host and grown in pure culture so that you can use that information. The pathogen must cause the disease when it is inoculated into a healthy host, and they must have the same signs and symptoms as seen in postulate 1. So you take your pure culture and you use that pathogen and inoculate a healthy host. When that infection occurs, you can look to see if they have the same signs and symptoms as seen in postulate 1. And then you can isolate that in order to examine it. The pathogen must be re-isolated from the new host, and then it must be identical to the pathogen in postulate 2. So they can use these postulates to trace back an infection to find its source. if romaine lettuce is causing infections, they can trace it back. They can examine all the pathogens from the people who are infected and trace it back to its source using Koch's postulates. We can classify infectious diseases based on certain things. Do the individuals who are infected have the same symptoms or the same signs? A symptom is a change in body function that is felt or experienced by a patient as the result of a disease. This is subjective. The patient tells you what they are feeling. They have a headache. They have a sore throat. Things that you cannot measure. It's subjective. They tell you what they are feeling. A sign is a change in the body that can be measured or observed as the result of an infection or disease. This is objective. You can measure temperature. You can measure blood pressure. You can measure respiratory rate. These are things that are signs. They have to be measurable. They are objective. Now, a syndrome is a specific group of signs and symptoms grouped together that accompany a disease or infection. A syndrome like irritable bowel syndrome or Down syndrome. Each of these syndromes have specific signs and symptoms so that you can identify that syndrome. A communicable disease is a contagious disease. It's spread from person to person. That is a communicable disease, a contagious disease. So a non-communicable disease would be a disease that is not transmitted from person to person. By understanding the occurrence of disease, we can understand how bad or how likely you might be to get a certain type of infection or a disease. It gives us a picture of how widespread this infection or disease is, or maybe it gives us a picture of its geographic location during a certain window of time. We can look at the incidence of an infection or disease. It's the proportion of a population that contracts a disease during a specific time. That time is usually determined to be during a year. These are new cases. Incidence only has to do with people who are newly infected, new cases. Prevalence has to do with the total number affected in a population at a given time. That's all cases. That includes people previously diagnosed and still alive that have this disease or infection. A sporadic disease is a disease that occurs occasionally in a population. It just pops up once in a while. An endemic disease means that it's constantly present in a specific population or geographical area. In the Pacific Northwest, we have a couple endemic diseases that are more prevalent in our area than they are perhaps if you compared them to the East Coast. MS and also meningitis have been considered to be endemic to the Pacific Northwest because we see more infections of these than in other parts of the country. An epidemic is a disease acquired by many individuals in a short period of time. This is often said to be for influenza. There will be an influenza or a flu outbreak in the city of Portland, and we will say that there is an epidemic of the flu in Portland. It's been acquired by many people in that population during a short amount of time. If it is worldwide, then as you know, and as we have heard and seen about in the news, It is a pandemic. A pandemic is a worldwide epidemic. Herd immunity is something that we really need to talk about when we talk about vaccinations. Herd immunity is the immunity in most of a population. This can occur when the population gets their vaccines, when they are vaccinated for different types of diseases and infections. If 90 to 95% of that population have those vaccines for those particular infections and diseases, we have a strong herd immunity, and we are able to protect those who are medically fragile or vulnerable, people who are not able to get vaccinations. Because 90 to 95% of the population is vaccinated, we are able to protect those who are vulnerable and cannot get vaccinations. Unfortunately, with a lot of individuals not vaccinating their children, our herd immunity is very low. It is not at a level where we can protect those who are medically fragile who can't get vaccinations. One estimate of the herd immunity in the Portland area was 82%, which is not high enough to produce a strong herd immunity for our vulnerable populations. When we look at acute diseases, these are diseases where the symptoms develop rapidly. You are sick right away. A chronic disease develops slowly over time, and it can last for a long duration. Subacute diseases, their symptoms are kind of between acute and chronic. This is not something that we consider in this class. Latent disease is a disease with a period of no symptoms. We talk about cold sores, HSV1, later on in the class. This is a latent disease. We could talk about shingles in the same way. These are diseases where you have a period of no symptoms, but you are still sick. Let's look at extent of involvement. Now, a local infection is an infection that's limited to a small area. Maybe you popped a pimple and you get a little bit of a staph infection in that area. Or maybe you have a stye. It's in the hair follicle near your eye. It's in a very small area. A systemic infection is throughout the body. It's circulating in the bloodstream, it's in the lymph, it's going throughout your whole body, and it can go to any area within your body. Now, a local infection can become a systemic infection, and that's called a focal infection. A systemic infection that began as a local infection. If you have a infection in your skin and it becomes bloody and scabbed over, that means that bacteria, that microorganism had access to the bloodstream, and it could potentially get in there and go anywhere it wants to in the body. The bloodstream and the lymph system, they're like highways that microorganisms can travel on. Bacteremia is bacteria in the blood. It's present in the blood. Septicemia is growth of bacteria in the blood. It's replicating. It's much worse than bacteremia. Bacteremia just meant that the bacteria was present. It did not mean that it was replicating and growing. Septicemia, though, means the growth of that bacteria in the blood. It does mean that it's multiplying, replicating, and potentially becoming much worse. Toxemia is the presence of toxins in the blood. A primary infection is an acute infection that causes the initial illness. You get strep throat. That's your primary infection. But you could have a complication with strep throat that can cause rheumatic fever or scarlet fever. But the primary infection is the first infection. It's the acute initial infection that causes the initial illness. A secondary infection is an opportunistic infection after a primary infection. It could be a nosocomial infection. It's an infection that occurs after you have the primary infection. Subclinical diseases have no noticeable signs or symptoms. The patient is asymptomatic. A lot of sexually transmitted infections have no noticeable signs or symptoms. There are certain predisposing factors that can make us more susceptible to infections and disease. Anatomy matters. Remember, we have all of those different portals of entry, but we're all unique in how our bodies are structured. Sometimes we are more susceptible based on our anatomy. We may have a genetic predisposition to certain types of infections or illnesses. These inherited traits are predisposing factors. Climate change and weather also have to do with making us more susceptible to certain infections and diseases. Because of climate change, our insect population has changed. We have more insects. We have more mosquitoes. Those can be potential carriers of infections and diseases. Our jet streams have changed, and things can get caught in the jet streams and travel for long distances. Fatigue matters in what kinds of infections we are susceptible to. When we're stressed, when we're tired, when we don't get enough sleep, our immune systems are weakened and we are more likely to become sick. Have you ever noticed that when it's final exam time, you might get a cold? You're more susceptible during that time when you are stressed. Our age matters. The very young are susceptible. The very old are susceptible. Age matters. Our immune systems might not be as developed when we're younger, and when we're older, we have comorbidities, and our immune systems have lost their strength as well. We are more likely to be susceptible to infections when we are older. Like in the case of COVID-19, it really has affected our elders in our population. Our lifestyle matters. What is our nutrition? Are we eating healthy? What is our socioeconomic level? Those who are in a lower socioeconomic level are more susceptible to infections. We don't seek treatment. We don't have insurance. We don't have the ability for good health care if we are in a low socioeconomic bracket. We may live in a food desert. We may eat junk food because those are the type of stores that are available to us. Drug abusers and those who are alcoholics also are more at risk to certain diseases and infections. Our lifestyle is very important to consider. Chemotherapy. Again, that's not just chemotherapy for cancer. That's other types of medications that we may take. if you have an autoimmune disease, you may be taking more toxic types of medications that are reducing your immune system, and you will be more likely to develop an infection or a disease. And again, stress. Stress can affect us. It affects our fatigue level. It also increases the level of cortisol, and cortisol is thought to be a complicating factor in a number of infections and diseases. So that's why all of these are predisposing factors and make us more susceptible to infections and diseases. There are many different types of reservoirs of infection. We are going to look at these three. We have human to human reservoirs of infection. STIs, sexually transmitted infections, are human to human cause. Things like HIV or gonorrhea. The carriers, again, may be asymptomatic when they have STIs. Or they also may be latent infections. They may have it and don't know they have it and transmit it to other people. Animal to human infections. Those are infections like rabies or Lyme disease caused from ticks. They are zoonotic diseases and are transmitted to humans from different types of animals. One theory on COVID-19 is that it was transmitted to humans at a wet market and possibly from pangolins that had been infected by bats. Those are zoonotic types of transmission, animal to human. Reservoirs of infection can also include things that are non-living, like the soil, like the dirt. Remember, endospores can survive in the soil, and they can cause both botulism and tetanus. We also have different types of disease transmissions. The contact can be direct, from person to person, which requires physical contact. Or it can be indirect, inanimate objects contaminated by different pathogens from an infected person or a reservoir. A fomite is an inanimate object like a desk or a table or a door handle. And if someone is sick and they cough into their hand and then they grab a door handle or they touch something, they are leaving those microorganisms behind on those fomites. And then you can come along and pick them up. That's an indirect contact. The person who was initially sick with the infection or the illness didn't give it to you directly. They didn't contact you, but you picked it up from something they touched or they contacted. We also have droplet transmission. Droplet transmission means that the infection, the bacteria, the virus is spread through the air in droplets. Sometimes they can go for 1 meter or less, and sometimes they can be even farther spread. A cough or a sneeze containing pathogens can spread in the air. This is direct contact transmission. The person is shaking the hand of another person. And if they had coughed into their hand or sneezed or went to the bathroom and didn't wash their hands, then whatever microorganisms are on their hands, they are now spreading directly to another person. This is an example of indirect contact. Again, someone went to the bathroom, they didn't wash their hands, they didn't clean them, and then they touched the door handle on the way out and left the microorganisms on the door handle. Then you came along and didn't know, and you touched the door handle to get out of the door, and you picked up whatever pathogens or microorganisms were left behind from the previous person. That's indirect contact. That's indirect contact on a fomite. This is droplet transmission. All of these droplets that can come out of your mouth when you cough or you sneeze can contain pathogenic bacteria or viruses. And sometimes, depending on the time of year, whether it's cold, whether it's hot, what type of virus it is, it can linger in the air in these droplets for an extended period of time. Disease can also be transmitted through vehicles or vectors, which we have talked about before. A vehicle is the transmission of a pathogen by an inanimate reservoir, like through food or water. That is a vehicle for the transmission of that microorganism into you. A vector, again, those are the disease carriers, those arthropods, the fleas, the flies, the ticks, the mosquitoes. Again, that mechanical vector is when the arthropod carries the pathogen on its feet. It flew into some fecal matter and picked it up, and then it flew along and landed on your sandwich when you were having a picnic. That's a mechanical vector. A biological vector carries the pathogen through some part of its life cycle inside of itself, like the mosquito carrying the protozoa through its life cycle in order to carry malaria from person to person. It's a biological vector. It carries it inside. That pathogen reproduces inside of that vector. That's a biological vector. A salad bar, even before COVID-19, was a vehicle of transmission for many different types of infections. From just someone coughing or sneezing on the food, you could transmit an infection. It also was a vehicle to transmit foodborne illnesses, foodborne infections from Salmonella or E. coli. People pick up the tongs from one type of food and transfer it to another type of food, or people putting their hands into some of the things on the salad bar and whatever was on their hands transferring into that food. Salad bars often had what was called a sneeze guard, like this plexiglass kind of barrier. But if you were short or if you were a child, you could sneeze or cough or spit or touch anything under that sneeze guard. So this is considered to be a vehicle of transmission. Nosocomial infections are hospital-acquired infections. They are secondary infections. Remember, we talked about primary infections and secondary infections. They are infections that a patient acquires when they go to the hospital that is not due to the cause of the first infection. 5 to 15% of all hospital patients acquire nosocomial infections. That is a terrible statistic. There are many reasons why nosocomial infections occur. There are a lot of vulnerable people who are in one place in the hospital. There's a lot of infection and a lot of diseases that have the potential to spread in that hospital environment. The hospital environment is often a place where it's super cleaned. So there's a lot of disinfecting going on. They really want things to be as clean as possible. Oftentimes, though, because bacteria are so adaptable, if you're cleaning and getting rid of the weaker type of bacteria, the ones that have not adapted, you leave behind the stronger, more virulent bacteria. the superbugs, as they're called. Those bacteria can then cause more infections. You got rid of all those little types of bacteria that can cause the smaller infections, but you've left behind these really strong type of microorganisms. Again, the people who come into the hospital have a lower immune system. They're compromised individuals. They are more likely to be susceptible to an infection, and so that can happen. And then there are many opportunities to have a chain of transmission. If someone has their catheter left in too long, they are at risk for urinary tract infections. Surgical procedures open up the body to the potential for an introduction of microorganisms into that area. There's many different reasons why nosocomial infections occur, but they shouldn't occur. These are hospital-acquired infections that the patient did not have when they went into the hospital. This is a table showing an example of some nosocomial infections that the CDC has presented on their website. Some of these things are really discouraging. Surgical site infections from an inpatient procedure. That is a very high number of patients who became sick with a hospital-acquired infection that they should not have received. Pneumonia. Pneumonia can occur from the use of ventilators. You're pushing air into the lungs of patients, and sometimes they can become at risk for developing pneumonia. But it's a secondary infection. It is not the reason why these patients went into the hospital in the first place. GI infections. That can just be because the healthcare professionals are not washing their hands and they're transferring fecal bacteria to the patient. UTIs, again, those can be caused by catheters being left in too long and the microorganisms are growing on the urinary catheters, causing these infections. Bloodstream infections, those are also usually parenteral. Remember, parenteral, it has to get in through a break in the skin. But once it does, it is able to get into the bloodstream and travel systemically. Here is a table showing different types of infections, gram-positive versus gram-negative. It could be C. diff or it could be a fungus. But what's interesting about the gram-positive or the gram-negative, you can see the percent of nosocomial infections caused by gram-positive and gram-negative bacteria. And then if you look at that percentage that's resistant to antibiotics, it's pretty alarming to me that some of these gram-positive bacteria that cause nosocomial infections can be 87% resistant to antibiotics. Our bacteria are evolving. Our bacteria are adapting. And when a patient who is vulnerable and immunocompromised is in the hospital, they are susceptible to these resistant types of bacteria, and it can be a very scary situation. If you work in healthcare, you might have encountered a patient who has had C. diff. C. diff can be a nosocomial infection. It can be hospital acquired. It is often caused by the overuse of antibiotics. The overuse of antibiotics can destroy the normal microbiome that protects the colon. And C. diff is able to take advantage of that situation and affect the colon, cause necrosis, cause the tissue to die. It changes the bowel function and it can cause very extreme diarrhea. 6,000 patients a year are infected with C. diff nosocomially. We are going to talk about C. diff again later on in the class, but for this purpose, we are looking at C. diff being a secondary opportunistic infection that has been acquired in the hospital. Klebsiella pneumoniae is another bacteria that can cause a hospital-acquired infection, a nosocomial infection. Remember, it has that protective capsule that it uses to stick to mucous membranes and to resist the phagocytosis of the immune system. It is caused, in this case, from normal fecal bacteria. So hand washing is the key to preventing transmission to patients. Healthcare providers must wash their hands so they don't spread this. It can cause pneumonia, surgical site infections, it can cause urinary tract infections, and it can cause a systemic infection of septicemia. It is spread through person-to-person contact, so this can be prevented. On this slide, you will see some of the reasons why infectious diseases are emerging. Things are happening that are causing more infectious diseases to occur, and you need to know this for quizzes and exams. There are many contributing factors. Remember, bacteria and viruses and many microorganisms are evolving. They're developing new strains. They are being adaptable to their environments, and so they are changing their normal structure. They're changing their normal way of functioning. They are evolving so that they are able to resist the immune system. They're able to resist treatment with antibiotics and removal from surfaces with disinfectants. They are changing and developing new strains. The inappropriate use of antibiotics and pesticides. We've talked about the inappropriate use of antibiotics. Please look back at previous PowerPoints if this is something that you don't remember or you don't quite understand. We are not using antibiotics correctly. We're overusing them. They're being prescribed for the wrong reasons. People are not taking their full course. There's many different reasons why it's inappropriate. The use of pesticides is also affecting the development of our immune system and making us more susceptible to these emerging infectious diseases. Climate change, again, has a huge impact. We talked a little bit about that. There's more insects. There's more mosquitoes. These changes, these climate change and changes in our weather patterns is causing more vectors to be around that can transfer infections. They can transmit infections. Modern transportation, air travel, has caused more infectious diseases to emerge. You can fly from one place to another and spread the infection, as we have seen. Ecological disasters, war, expanding refugee camps, human settlements. When governments are not stable and there are crises in those countries, then it is more likely that infectious diseases will develop and they will spread from person to person because there is not a healthcare structure to help prevent and treat these infectious diseases. public health failures. There are times when public health does not recognize a situation and an infectious disease is allowed to take hold and spread before it is understood and treated how it should be. Let's look at some review slides. How would you characterize the human microbiome? Is it A, commensal, B, parasitic, C, mutualistic, or D, both B and C? Is it both parasitic and mutualistic? The answer is D. Yes, it is absolutely mutualistic, which means we benefit from our normal microbiome. It can protect us if we have a healthy normal microbiome from pathogens being able to take hold on or within our body. However, it can also become parasitic and it can be opportunistic and cause infections or diseases to occur in our body when our immune system is not working at its best, when we're under stress, when we're tired. It can become opportunistic. So yes, it is both B and C when we talk about the symbiotic relationship. The study of the cause of disease is called A, etiology, B, pathology, C, epidemiology, or D, microbiology. The answer is A. Etiology is the study of the cause of disease. Normal human microbes can be pathogens. A, true, B, false. The answer is absolutely, unfortunately, A, true. Our normal human microbiome can become pathogenic and can become opportunistic. Usually it's great and usually it helps our immune system, but sometimes under certain conditions, it can become pathogenic and be opportunistic and cause infections. Which of the following is sterile? Our eyes, our GI tract, our lungs, our cardiovascular system, or E, none of the above? The answer is E, none of the above. We used to think that there had to be parts within our body that were sterile, that no microbes could get to. But unfortunately, through evolution of microorganisms and the increase in their virulence factors, there really is no place within our body that is sterile, because microorganisms can reach all areas of our body and enter our body and use them as portal of entries, like our eyes, in order to spread throughout the body. So none of the above. E. In 2017, 2.4 million people were newly infected with HIV, and 34 million people were living with HIV and AIDS. The prevalence of HIV is A, 2.4 million, or B, 34 million. The prevalence is total cases, all cases. So the answer is B, 34 million people. That's the prevalence of HIV. B is the answer for this question. If we were talking about incidence, that's new cases, and that would have been 2.4 million people. That would be incidence, but we're talking about prevalence, which is the total number of cases of individuals who have HIV or AIDS, and that's B, 34 million. A patient with herpes simplex virus 1 tells you they have not had a cold sore for months. HSV1 is A, an acute disease, B, a chronic disease, C, a latent disease. The answer is C, a latent disease. HSV1 can have a period of time where the individual who is infected with the virus does not have signs and symptoms. That would make it a latent disease. You can go for months without a cold sore, and then all of a sudden you have something that stimulates and activates that virus to start replicating again because it's been integrated into the genome. It can go into that lytic cycle, from a lysogenic cycle to a lytic cycle, and start making more of the virus and cause a cold sore to occur. But HSV1 is a latent disease because you can have a very long time without signs and symptoms when that virus is in the lysogenic stage.